Today's lecture, we're going to review control of the cell cycle. You've probably seen parts of this in other classes, but we'll get into a little bit of detail of the important regulatory proteins involved in controlling the different phases of the cell cycle. Just to remind you, the cell cycle are sequential events or phases to help prepare a cell for division. A cell that's going to enter into the preparatory stages of division will first start in G1, where a cell will specifically grow and prepare for S phase. Then the cell will move into S phase. S phase is for DNA replication or duplication, followed by G2, in which the cell will grow and additionally and prepare for mitosis. In M phase being mitosis, the sequential phases of nuclear division or DNA division. The cell cycle lengths can range from about 30 minutes in a frog embryo, which actually has an absence of G1 and G2 phases, or it can be actually up to hours to days for this division process to occur, such as in the liver, which is months to up to like a year for a cell division process. When we think about M phase and what happens, keep in mind we have our specific phases of mitosis: prophase, prometaphase, metaphase, anaphase, and telophase, followed by cytokinesis. So that ultimately you end up with two daughter cells at the end with equal number of chromosomes. Now, as a reminder, keep in mind with the cell cycle, starting with G1 into S and G2, that is what is called interphase for a dividing cell. And the M phase is the division phase when we think about mitotic chromosome separation. Now, when we think about points of control and how cells are regulated during this sequential phases for preparation of cell division, there's three basic control points. The first one being what's called at the end of G1, the G1 checkpoint, and important assessments that occur at this particular checkpoint. Is the environment favorable? Are the chromosomes healthy? And if all those important factors are present, the DNA is healthy, then the cells will progress forward through S for DNA synthesis. Then the cells will progress through G2, and there's another important checkpoint, which is called the G2 checkpoint. Has the DNA been replicated effectively? Is there DNA damage? If any of these things have not occurred as far as the completion of DNA replication or DNA is damaged in a normal healthy cell, that will initiate what's called the G2 checkpoint, and essentially the cells can pause until appropriate actions are made to repair the cell or complete the proper events needed for M. There's also a third checkpoint in mitosis, which is called the metaphase anaphase transition. And this is controlled by another protein complex, which we'll talk about in a few minutes. But ultimately, this is critical for proper separation of the DNA into two separate masses so that ultimately each resulting daughter cells end up with equal amounts of DNA. So some time ago, it was actually in the early 70s, late 60s, early 70s, it was thought that there had to be some sort of factors that would help stimulate cells to enter into mitosis. And there was two scientists, which are shown here, Rayo and Johnson, that actually published results really identifying the fact that mitosis required specific key factors to initiate key events of mitosis, so for a cell to enter into mitosis. And what they did to prove that certain factors or determinants were important or required is they actually fused two different cells together. They fused mitotic HeLa cells with another cell type, which rat kangaroo, PTK2 cells, that are at different stages. And so this particular diagram here shows what happened when an M phase cell was fused with a G1 cell, the PTK cell. And what happened was they saw premature condensation of the G1 chromosomes. So you can see the mitotic chromosomes from the mitotic cell, but here the G1 chromosomes actually starting to condense as well. So there had to be some factor that was present in the mitotic cells that led to condensation of premature condensation of the G1 cells. They then repeated this same type of experiment, but then fused these M phase, these mitotic HeLa cells, with PTK cells that were also in G1 and also in G2, which the G1 we just saw. With the G2, it also, there was some factor in the mitotic extracts cell that caused early condensation also of the G2 chromosomes, which you can see here. But they did note one difference, one of which is there were 2 copies of these chromosomes in the G2 cells, which would indicate that DNA synthesis has occurred and we have our sister chromatids attached, which was different than the G1 because there was no DNA duplication yet. They also found that the G1 the S phase cells were fused with the mitotic M phase HeLa cells. It actually caused premature condensation, but it also caused the DNA to fragment because the chromosomes were undergoing DNA replication. And so ultimately that caused kind of a fragmentation of the DNA and condensation of those pieces due to the current process of those chromosomes being replicated. Regardless, these experiments really provided the fact that mitotic cells had some sort of factor that led to stimulation of mitosis. And of course, the next sets of experiments that proceeded after this was, what is this factor that leads to initiation of specific phases of the cell cycle, and more specifically, entry into M phase. And many studies using Xenopus oocytes or other types of invertebrate embryos led to the identification of what's called the MPF factor, and maturation promoting factor. And this MPF factor or complex was ultimately required to initiate mitosis in something like Xenopus oocytes or other invertebrate embryos. And it's further been shown in other types of mammalian cells as well. MPF, when it was analyzed, consists of 2 subunits, one of which is a kinase we now call CDK, cyclindependent kinases, and another protein, a regulatory protein called cyclin. So it has what's called CDK and cyclin as this dimer complex. And what they also found was MPF activity was high during mitosis, at the onset of mitosis. So it helped highlight the fact that the activity of this complex helped trigger the entrance into mitosis. What they also found was the amount of cyclin, which interacts with this kinase that forms the complex, also had peak levels when this complex was most active. Cyclin, which was hence its name, cycles in concentration and builds up to a peak point so that more complex can form, which will lead to increased activity. And the increased concentration of this cyclin protein helps activate the complex. We now know, after studying the MPF factor as well as now many different cyclin-dependent kinases and cyclin complexes, most of which, some of our most important studies of these complexes, these MPF-like complexes, which are cyclin-dependent kinases and cyclin, most of the work was done initially in yeast. Yeast is a really good model system. It's simpler. It's a simpler eukaryotic cell. And what they found was, looking at yeast cells, was there was a kinase in cyclin complex that 2 different complexes, in fact, that were critical for progression through the cell cycle of yeast. One complex, the CDC2 kinase and the G1S cyclins, was critical for passage through the G1 checkpoint. And there was another second complex also including CDC2 kinase, but different mitotic cyclins that were important for the onset of mitosis. So ultimately what they discovered in now yeast, and we're going to get into higher mammals in a second, were that these CDK cyclin complexes were critical for the G1 and G2 complex. G1 and G2 checkpoints and progression through. So their activity is required to actually move the cell from G1 into S or G2 into M. And in this particular example, in fission yeast, there's one CDK that's used for both checkpoints, CDC2 kinase, but different cyclins associate with this CDC2 to activate the specific activity needed at the G1 checkpoint and also at the G2 checkpoint. So this is at the G2 checkpoint, and this is our G1 checkpoint. So keep in mind, to progress through the cell cycle so a cell sequentially can go through all those key events to move into division, it requires key activation of these kinases so that ultimately these kinases will allow for their activity to help trigger key events to move into those next events. So keep in mind, remember, kinases phosphorylate things, and their activity will lead to phosphorylation of key targets, which will trigger the next event. So how is this activity controlled so that only specific events are triggered in their proper order when the cell's healthy and the conditions are correct for that division process? So here shows you, this particular diagram shows you the important key events that lead to activation of these CDK cyclin complexes. Like I said, that are going to be important for allowing a cell going from G1 into S at the G1 checkpoint, or allowing the cell from going to G2 into M at the G2 checkpoint. So the activity of these complexes are critical for progression through the cell cycle. And this is one example. This was shown by many different mutant studies looking at yeast. And the CDC2 kinase is inactive. And one of the first steps to its activation is it binds to a specific cyclin. There's high enough levels of that specific cyclin that essentially are going to allow for complex formation. Additionally, 2 phosphorylation events will occur to the CDC2 kinase, one of which is at the threonine 161, and the second is at the tyrosine 15. Now, the kinases that add these phosphorylation events are shown here. CAC phosphorylates at the 161, and we phosphorylates at the tyrosine 15 residue, which is shown here. Now, what you'll notice is the CDK and its cyclin with both these phosphorylations is still inactive. And full activation of this complex actually requires removal of the tyrosine 15 phosphorylation group. And that is done by a phosphatase called CDC25. It is at this point in time that the CDK cyclin complex will become active. And you can see that this dephosphorylation event happens in fission yeast at the end of G2, which will help trigger the activity of the CDK cyclin complex to initiate mitosis, which you see here. Now keep in mind, once mitosis is complete, this complex needs to be inactivated in this particular scenario. And one of the inactivation steps is actually to degrade, destroy the cyclin protein so that the complex cannot no longer form. So these 3 kind of key events are needed. 1, there needs to be enough cyclin. 2, we get phosphorylation of both an activating and an inactivating site. And 3, for activity of this complex, there has to be removal, the inhibitory phosphorylation on the CDC2 kinase complex. Now this also shows you, this next slide shows you other important points of control for CDK regulation. One of which is there has been CDK inhibitors that have been identified that essentially can inhibit this activity. This is important if certain events or problems happen during these sequential events. Certain inhibitors can be activated to inhibit the activity of the CDKs, a specific type or specific types of CDKs, so that progression through the next phase does not occur. One example protein that was discovered in yeast is called SYK1. You're also going to see another example which is called P21 and also another example called P27 in a few slides from now. And another way of control of this activity, as I suggested from the other slide, is the fact that the cyclins also can be degraded so that the complex no longer is associated. And this degradation ultimately can lead to inactivation of the complex itself. And this degradation is promoted by ubiquitination and destruction by proteasomes. And there's different types of ubiquitin ligases such as SCF, or what we also call APC, which you're going to see a little bit later, which help remove or site those protein cyclins for degradation. Another mode of regulation of CDK activity besides inhibitors and destruction of cyclins is also different movement of cyclins to different locations so they no longer can effectively bind to a CDK. For example, prevention of movement of the important cyclin B out of the nucleus, not allowing it to enter into the nucleus, will actually prevent activation of the CDK that's important for M phase initiation. So different locations in which these cyclins are found, the cytoplasm or the nucleus, can help control whether they can interact with their specific CDK at a certain location, which can control activity. So since the discovery of CDC2 in yeast cells, the search for different CDKs have also been pursued in different mammalian cell lines. And this particular diagram shows you what has been discovered in reference to CDKs and specific cyclins and in mammals. And what they find is, unlike yeast that have one CDK, which is called CDC2, which happens to be in fission yeast as Pombe, they have found that mammalian cells have more CDKs and more cyclins, which you can see here. So progression through G1 and initiating S phase requires CDK4 and 6 plus the D cyclins, and there's multiple types of D cyclins. Also, progression further into S phase requires CDK2 and cyclin E. And further progression into S phase requires CDK2 plus A cyclin. And then also we have a mitotic initiator CDK, which is CDK1, and it complexes with B and A to help stimulate M phase. So that's what's shown here. So there's multiple CDKs and cyclins and specific complexes that form with specific CDKs. And that's what this is highlighting down here. So the pairing that they see, what CDK and what cyclin it interacts with, is highly specific, and only certain combinations are found. And that combination will allow for interaction with key substrates, specificity for key substrates, so that it helps trigger the next phase. So to highlight the significance or importance of these CDKs, there has been knockout experiments of each one of these CDKs in mice to really assess significance. And that's essentially always what knockout experiments are. When you knock out a gene, is it lethal for development of the mice? And what you'll see from this particular diagram is only one CDK and its knockout is considered lethal, meaning that essentially it doesn't even allow the embryo to start to form, and that is CDK1. And CDK1 is critical for especially mitotic progression, so entrance into mitosis, but it appears to be critical for driving the mammalian cell cycle. However, when they looked out at other knockouts of different CDKs, you'll notice that knockout of 4 and 6, where CDK1 and 2 are present, that you do get progression of development of the mice, but you can see that they actually don't live past birth, which is kind of indicated by the visual view of the mice. However, what they did notice was Also the same with CDK2 and CDK4. Both of these two different knockouts, as I said from before, the 4 and the 6, both led to development to occur, but did not allow for those mice to actually survive post-birth. There was one combination that did allow for the animals to survive, however, they were sterile, no longer able to reproduce, and that was the CDK2 and CDK6 knockout. Essentially, CDK1 and CDK4 were enough to allow for almost proper production of that organism, lacking the fact that they were able to reproduce. So what does this all mean when we think about the mammalian cell cycle? The CDK1 appears to be the most essential, and it appears to have the different CDKs appear to have some overlapping functions, which CDK1 appears to compensate for somewhat. So even though we have all these different CDKs, CDK1 appears to be the most essential due to the loss of it and how lethal the loss of CDK1 is. There was also some important knockouts of cyclins, which is shown here. And same was true, some of the others knockouts. If you knock out one, some of the others would compensate for that knockout cyclin as well. So there's some overlapping function, and it appears that some of them may be able to phosphorylate similar targets ultimately, which would allow for progression through the cell cycle. So ultimately, progression through G1 into S requires specific G1 and S CDKs. So essentially the activation of CDKs so that the cells can move into S phase. So let's talk about the first checkpoint, the G1 checkpoint. A cell is going to move through G1 and essentially then move through into S phase. To move into S phase, it requires activity of specific G1S CDKs, specifically when we think about mammals. And what are these CDKs doing to help stimulate the next phase? What is their activity and how is that important? So there's been several important targets that these CDKs interact with and phosphorylate that help trigger the next event. When we think about what leads to the activation of these CDKs, so they can phosphorylate key targets and we get cell cycle progression, there has been shown to be specific signal pathways activated that leads to activation of these CDKs so that the specific kinases phosphorylate them. And then we also get activation of the CDC25, which is the phosphatase to remove the inhibitory phosphate. And that's critical for getting activation of these CDKs so that they can allow for specific targets to be phosphorylated and we get progression forward. One such example are mitogens, growth factors. So if a cell is in G1 and it's receiving growth factors, we have receptors that can bind to growth factors. It will help lead to signal pathways that help trigger the activation of those kinases to help phosphorylate the CDKs so that it can start the events on activation of the CDKs so that essentially the cell can move forward, which may make sense if the cell's receiving growth factors, there's surrounding cells that are telling it that it needs to divide. And this will help kind of stimulate that process of cell division. So after looking at these CDKs, and specifically the G1S CDKs that are important for stimulation of S phase, they've found different targets that these CDKs interact with that allow for progression or start to allow for progression into the next phase. One important target of the G1S CDKs is PRB. PRB normally inactivates an important protein called E2F, which is shown here in green. When the CDK is active, which you can see this active complex and what led to its activity here, it will phosphorylate PRB, and PRB, when it's phosphorylated, will lead to release of a transcription factor called E2F. E2F, when it is released, it will bind to specific genes and lead to their transcription. And some of the genes that become transcribed in the presence of E2F are histones, DNA polymerase, and important enzymes needed for nucleotide synthesis, etc. So you can see here. So ultimately, phosphorylation of PRB by these G1S CDKs will help initiate or start S phase by leading to production of key proteins needed for DNA replication. So DNA polymerase, histones, etc. So this is an important target to allow for S phase to start. We have to have the machinery present for S phase to be initiated. Another important target that's been identified for the SCDK when it is active is both the CDC6 and CDT1 proteins, which we heard a little about last week in one of our presentations, and also phosphorylation of what's called the origin recognition complex, the ORC. Now, keep in mind, we talked a little bit about the fact that we have these specific origins where DNA replication is initiated, and there's special proteins that allow for recognition of these sites so that the machinery is actively recruited to these sites. Some of these important complexes that have been discovered both in yeast and mammals is the fact that there's what's called a origin recognition complex, which is shown here, and there are different proteins that actually bind to this complex, one of which is CDC6, and there's another important protein called T1, and it helps bring what are called MCMS complexes to the site. Now, these complexes will bind in G1, and initiation and binding of these complexes are triggered essentially by kind of stages through G1. However, SCDKs will phosphorylate these proteins and essentially help initiate DNA replication. So we get phosphorylation of CDT1, and we also get phosphorylation of CDC6. That helps remove CDC6 from these origins and also helps remove CDT1, which you can see. CDT1 gets degraded and CDC6 actually gets removed out of the nucleus so that it can't interact with these complexes. And that will help initiate DNA replication. Now, kind of the two important events to this phosphorylation by the SCDK to these different complexes of the origin recognition complex, the CDC6, the origin itself, which you can see here, and also CDT1, is the fact that it triggers initiation of DNA synthesis, and removal of CDC6 and CDT1 actually prevent re-replication, so that DNA replication only happens once. So keep in mind, you've learned about these CDKs, and their activity is critical for movement from G1 to S, and there's also CDKs that are important for movement from G2 into M. And right now we've just talked about what are some of the activities of the CDKs that are critical for the G1 checkpoint, and how is their activity important for moving the cells into something like S phase. And we've looked at two different targets now, PRB and CDC6 and CDT1. Now, what happens if we have something that happens to the cell that the cell should not progress through S phase, let's say, G1 to S phase? If there is DNA damage, this will allow for cell cycle arrest, or the cell will arrest at the checkpoint, either the G1 or the G2 checkpoint. And different damage to the DNA will help initiate this cell cycle arrest. And there's different complexes that ultimately will sense different types of DNA damage. If there's double-stranded breaks, there's a complex called ATM, which will become activated, and it will then, by binding to certain complexes at this double-stranded break, it will lead to activation of CHK2, which is a kinase, and this leads to phosphorylation of P53. This leads to the stability of P53, meaning we'll get increased levels of P53. And P53 is a transcription factor, and it will bind to one gene called P21 gene, and that will lead to expression of the P21 protein by transcription translation. And P21 will then bind to the CDK and physically inhibit its activity. So P21 is a steric inhibitor of the CDK. So this is important if for example, a cell was damaged, it had double-stranded DNA breaks, maybe by ionizing radiation, then essentially that would lead to increased levels of P21 protein. It can physically bind to the G1S CDKs and inhibit their activity, so they cannot phosphorylate targets like PRB or CDC6 to initiate DNA replication. We don't want to move into S phase if we have damaged DNA. This can arrest the cell until the damage is repaired, or if the cell can actually be repaired, it can actually help stimulate apoptosis or cell death of the cells. Now, we also have another type of damage pathway that's initiated by what's called ATR. This is initiated due to different types of DNA damage, one of which is if there's a stalled replication fork during DNA replication, or there's other types of damage as well. And ATR, what it will end up doing is activating CHK1, and CHK1 will phosphorylate CDC25. If you remember right, CDC25 is that important phosphatase that's needed to remove the inhibitory phosphate on the CDK. And removal of that phosphorylation leads to activation of CDK. Well, how does phosphorylation of CDC25 influence activity of CDKs? And what it does is it inactivates it so that it can't interact with the CDKs so that they become active. So it prevents activation of CDKs by inhibiting that CDC25 activity, that phosphatase activity. And it prevents it because it cannot now, that inhibitory phosphorylation on the CDK cannot be removed, so it leads to cell cycle arrest as well. So two different pathways that can be initiated depending on what type of DNA damage occurred that essentially will lead to inactivation of CDKs, which prevents cell cycle progression into the next phase. Now, when thinking about different types of inhibitors like the P21, there's also an inhibitor called the P27. And P27 is very similar to P21. And P27, different structural analysis studies, such as X-ray crystallography, have shown P21 and interaction with something like CDK, the cyclin A complex, and binding to that CDK, allowing for inhibition or steric hindrance so that it can't interact and phosphorylate key substrates. So ultimately, something like P21 or P27 are going to essentially bind, interact with the CDK complex, and not allow them to interact with key substrates so that they can phosphorylate them. So essentially blocks CDK activity and cell cycle progression. So it allows for that cell cycle to be arrested. Now, there have been different studies looking at the activity of P27, this inhibitor that's potentially this protein that serves as an inhibitor for CDK2. And what they found is P27 seems to be significantly important during development, and expression of P27 helps allow for a cell to stop dividing and become differentiated during development. So it essentially allows for decrease in cell division and differentiation to occur so that ultimately different morphological features of cells can occur during embryogenesis. And in fact, if they eliminate P27, they do a knockout of P27 in mice, they find something very interesting. The P27 knockout mice is brown, and a normal control wild-type mouse is in the white. And what you notice is they get this extra large mice in the absence of P27. And in fact, when they look at different organs of that knockout P27 mouse, they find that something like this is just showing you the thymus. The thymus is much larger in the knockout mice than the control. What does this mean? P27 is critical for proper development such that it prevents further cell division of cells. And in fact, if you inhibit its function, then you're going to increase cell division numbers, which leads to an increased size and mass of organs, which you can see from here. Okay, so let's talk about the G2 checkpoint. and specifically what those CDKs do to help go from G2 and initiate M phase. So we've talked a little bit about the G1 checkpoint and the G1 and S CDKs and what they might do to help progress a cell into S phase. Let's talk a little bit about the M phase stimulation. So the CDK1 in mammals is the important CDK that is critical, its activity for progression into M phase from G2. So after much analysis of the CDK1, the MCDK for mammals, or this what was also called the CDC1 in yeast, that they found important targets also of this CDK that help stimulate mitosis. And some of these are going to sound pretty familiar. One of which is phosphorylation of histone 1 proteins. This important event is thought to help lead to condensation of chromosomes, which is critical for the beginning of mitosis. Another important target is phosphorylations of lamins. So the MCDK phosphorylates lamins and other proteins associated with the nuclear envelope, and that helps stimulate nuclear envelope breakdown. And another target that's been shown as well is phosphorylation of RNA polymerase. And phosphorylation of RNA polymerase is thought to lead to its inactivity. And this is important because we're condensing chromosomes. It makes it really difficult to actually get at the gene structures, and therefore we don't want transcription occurring at this time due to this highly condensed structure. So phosphorylation of RNA polymerase is thought to lead to inactivation of RNA polymerase to reduce transcription at this period of time during division. Now there's also a third checkpoint. So we talked a little bit about the fact that we have a G1 checkpoint and the G2 checkpoint, both of which are controlled by the activity of CDKs, meaning that CDK activity, specific CDK activity, will allow for progression through G1 into S and also G2 into M. But we also have a third checkpoint, and this is the spindle assembly checkpoint or the M phase checkpoint. And it's also called the metaphase-anaphase transition. And ultimately, this specific checkpoint allows for proper segregation of chromosomes. And essentially, chromosomes need to be aligned at the metaphase plate during metaphase before anaphase will be initiated. So that's why it's called the metaphase-anaphase transition. So it's a key event that will not allow chromosomes to separate unless they are properly aligned. So what happens is chromosomes, unattached kinetochores, essentially chromosomes that do not have their microtubules properly attached to one side, so improper attachment or alignment, meaning that the chromosomes are not truly aligned in the middle, these chromosomes will send a weight signal. And this weight signal is basically by production of a certain complex called the MCC complex. And this suppresses the activity of a key protein called APC. So if the chromosomes are not aligned, then essentially the APC will not be activated, and therefore you do not get initiation of anaphase. Only when chromosomes are aligned will the weight signal stop being released, and then we'll get activation of the APC, and then that will allow for initiation of anaphase. Now, this is a really critical checkpoint because it ensures that there are a proper number of chromosomes that end up in the resulting daughter cells at the end of mitosis. Alignment allows for proper segregation and proper numbers of chromosomes. So ultimately, the APC is the important complex that's needed to help initiate anaphase, and this will not be turned on until true alignment has occurred. So this is showing you here during metaphase, the chromosomes will move back and forth due to proper attachment to microtubules and ultimately end up aligning. And this alignment is thought to be due to equal tension from either pole, and that usually implies equal number of microtubules attaching to the kinetochore region of those chromosomes. If a chromosome is not aligned, which is shown in this picture, the APC will be turned off. However, if alignment occurs, then the APC will be turned on, which will lead to sister chromatid separation during anaphase. So what is this checkpoint? Excuse me. Unattached chromosomes, or kinetochores, lead to formation of an important complex called the MCC. The MCC stands for mitotic checkpoint complex, and the activity of this complex that forms on these unattached chromosomes actually blocks APC activity. So in this particular diagram you're seeing here, this chromosome is unaligned, and that leads to formation of this complex. This complex has a few what are known as kinetochore proteins and checkpoint proteins, and it's BUB-R1, BUB2, MAD2, and CDC20, which you can see here. This complex is important because these three proteins hold an important protein called CDC20. CDC20 normally, if it's released, will bind to the APC, and it's an important substrate or subunit of APC that leads to its activity so that it can actually lead to sister chromatid separation. But if we have an unattached kinetochore, CDC20 will bind to this MCC complex and will not be able to interact with the APC. However, once these chromosomes are properly aligned, which you can see here, CDC20 will be released because this complex will dissolve. And when CDC20 is released, it binds to the APC and allows for now interaction with key targets. One important target is destruction of a protein called securin. And securin holds separase. And when securin is released, the separase is an enzyme that will cleave cohesin rings that hold sister chromatids together, thus allowing the sister chromatids to separate during anaphase. So ultimately, this MCC, this mitotic checkpoint complex, inhibits the APC by binding to CDC20, and essentially that's going to prevent activation of APC and sister chromatid separation until CDC20 is released, and it's only going to be released when alignment occurs. So this next diagram highlights the role of APC. So normally, separase is inhibited, this enzyme is inhibited by a protein called securin. Activation of APC when it binds to CDC20 will lead to destruction of securin. And that's because APC is a ubiquinase ligase, and it ubiquinates it, so it adds a modification that selectively gets it destroyed by proteasomes so that it's destroyed. So you can see that now it's degraded. This will then lead to the separation of securin from separase will lead to activation of separase, and separase will cleave the cohesins, the rings that hold the sister chromatids together, and this will allow for sister chromatids to separate, which will help initiate anaphase. So this completes our cell cycle review, specifically cell cycle control. So just keep in mind there are three different checkpoints: the G1, the G2, and the metaphase-anaphase transition checkpoint, all of which are controlled by specific proteins, and specifically to progress through those checkpoints, their specific activity of certain proteins is required. So for the G1 and the G2 checkpoint, there are specific CDKs that their activity are required to help progress the cell forward into the next phase. And they'll have key targets that they interact with and phosphorylate to lead to stimulation of that next phase. So in G1 and G2, it's CDKs, and in the metaphase-anaphase transition, the mitotic checkpoint, it happens to be APC. And metaphase alignment is critical for activation of the APC. We also have key proteins that can inhibit these CDKs, specifically such as G1 and G2 checkpoints, if certain circumstances arise, such as DNA damage. lack of nutrients, etc. And essentially that will help lead to inactivation of these CDKs so that cell cycle progression is halted, so the cells will arrest. So ultimately, proper movement through these cell cycle phases is tightly controlled so that cells will divide properly and also provide healthy DNA to both daughter cells at the end. There's also a paper posted on looking at regulation of CDC and its role in initiating DNA replication and preventing re-replication, and what happens to CDC6 to allow for the initiation of DNA replication and preventing re-replication. So that is the supplemental paper for this particular lecture. And there's also questions posted for that specifically highlight important parts from this paper that I want you to address.